right. This lecture is now being recorded. Yeah, Khan Academy should be your top review source besides, AP Classroom. Khan Academy is pretty much the gold standard for any sort of subject review. All right, well, I think it's about time that we got started with the lecture. So, once again, this is the AP Biology full course review. So, welcome, good evening, let's get started. So, a bit about this lecture. This review session was prepared by Golden, that's me, as part of Five Hive. We are a student-run initiative with over 70 volunteers and 10,000 plus combined hours, and what we do is we provide student-created resources for students taking AP courses, which is you guys tonight. You can check out our website at fivehive.org for additional AP resources and practice exams. If you'd like to contribute to us by creating resources, hosting lectures, or promoting this project, you can check out the document, which is linked in the slideshow for more information. And below you can find our socials, our Instagram, TikTok, link to our Discord, and our website. And if you guys would care to fill out the survey regarding the lecture by either scanning this QR code or typing in this bit.ly link, it'll ask you just a couple of questions about this lecture, what you would like to see, things like that. Your feedback is greatly appreciated. Not only is this going to help me improve my lectures, it's also going to help students in the future who take this course. All right, and as I previously said, this presentation, the lecture, I don't intend for this to be a comprehensive guide to every single thing you've learned in the course, but you should treat this as more of a review session. What that means is I can't possibly get into minute detail about every topic covered throughout the exam, so I've picked what I think are the most important topics and concepts for you to review. And that being said, I also can't guarantee any specific score on the exam, which is May 5th at 8 a.m. local time, or any particular grade in the corresponding high school course. And AP is a trademark registered by the College Board, which is not affiliated with and does not endorse this resource or the Five Hive team. And finally, just to reinforce your understanding, make sure you get the most out of attending this lecture today, I do ask that you please engage with the lecture, which means asking questions in the channel chat, which I will check periodically, participating in practice questions, and also discussing the lecture content with each other. So let's get started. An overview of what we're going to cover tonight. First, we're going to get a quick understanding of how prepared each of you are for the exam. Then we're going to run through each unit. We're going to look at what topics the unit entails, and then I'm going to lecture quickly about the unit. And then after that, after each unit, there will be one or two quick progress check style multiple choice questions where you will see the question on the screen, you will see the answer choices, and I ask that you reply with the answer in the channel chat. And then after that, we're going to go over, at the very end, we'll see places where you can find additional review material, and then I'll open up the stage to any questions. All right, so first, let's just take a moment, gauge your own readiness on your knowledge of AP Biology, the entire course. All right, so it looks like, based on our answers so far, the average looks to be about a 5 or maybe a 5.5. It's a 6 range. So at the end, I hope that's going to be somewhere closer to the 8 or 9 range, maybe even some 10s. So that being said, let's get started. Unit 1 is about the chemistry of life. You'll see that it's about 8 to 11% of your AP exam, and it includes these following topics. So first thing you should have learned about in this course is the structure of water. Water is a polar molecule, and it can interact with other water molecules via hydrogen bonding. And the water has the following properties that are basically make life possible on Earth. Adhesion and cohesion, which is water sticks to other water molecules as well as other substances. Water has a high specific heat and heat of vaporization. It has a lower density in its solid form, which is ice. So basically, the saying ice floats on top of water as opposed to sinking underneath it. And water is the universal solvent. You can see on the right here, we've got a little diagram of water. You can see its asymmetrical structure makes it polar, which means which gives it these following properties that we've just gone over. And then the elements of life, three main ones that you should know are carbon, which is by far the most abundant, and then is nitrogen, phosphorus. And if we look on the right here, as I've said, all life forms are carbon-based. 99% of the human body, and to an extent, all life on Earth, is made up of the following six elements, not in any particular order, but they are oxygen, carbon, hydrogen, nitrogen, calcium, and phosphorus. And you should have learned that organisms must exchange matter with the environment to grow and reproduce. And regarding these three most common elements, carbon is used to build biological molecules used in cell formation. Nitrogen builds proteins and nucleic acids, which is your DNA and RNA. Phosphorus is also used to build nucleic acids and lipids. Regarding the content of the exam, you don't need to know all this different stuff. Like, you don't need to know the physical state, crystal structure. That's more AP chemistry stuff. You just need to know carbon, phosphorus, nitrogen, and what they're used for in the body. All right, and then next up is macromolecules. For the scope of this course, there are four main ones that we're interested in. Those are carbohydrates, proteins, lipids, and nucleic acids. Each one has its own special building block called a monomer. You can see them in the second column here. The point here is that monomers combine in long chains to form the polymer, which is the macromolecule. And over on the right here, you can see the function of each of these macromolecules. And then for the sake of time, I won't go into too much detail about each one, but you can definitely look through the lectures slides on your own time to kind of like catch up on things that maybe you missed. So nucleic acids, as I previously said, these are your DNA and RNA. They are composed of nucleotides, which is the monomer. There's a 5' end and a 3' end. And when we synthesize DNA and RNA, we always add nucleotides onto the 3' end. Typically, DNA has two strands of nucleotides running antiparallel. That means they run in opposite directions in a double helix, so they spiral around each other pretty much. And then the bases bond to each other. The bases only bond to specifically other bases, where adenine A bonds to thymine T. And that bond has 2 hydrogen bonds in it. And then guanine bonds to cytosine using 3 hydrogen bonds. And the proteins are long chains of amino acids, of which there are 20 different amino acids. And for proteins, the order of the amino acids will determine how the protein folds locally. And this will influence the overall shape of the folded protein and how it interacts with the other proteins around it. And what influences and you might be asking, well, what influences how a protein folds? These are things like charge of the amino acids, things like that. And the protein will always fold in a manner as to minimize the chemical potential energy in the different amino acids interacting with each other, which is what is summarized out here. We're always trying to achieve the most stable form of a protein, which is what defines how the protein is folded. And then carbohydrates, otherwise known as carbs, are chains of monosaccharides, which is just a fancy way of saying simple sugar, connected by covalent bonds. They can be linear or branched, which just refers to its molecular structure. And carbohydrates are always composed of carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. So for every carbon atom, there's going to be 1 oxygen atom and 2 hydrogen atoms. And that's how you know it's a carbohydrate. So if you look at glucose, you see it's the chemical formula for glucose is C6H12O6. And there's your 1 to 2 to 1 ratio. And finally, we have lipids, which are just long chains of fatty acids also connected by covalent bonds. Lipids are nonpolar. And we can see down in this diagram that we can have both only single bonds in the case of saturated fats, and in unsaturated fats where we can have double bonds. which creates a slight bend in the molecule of a lipid. And then a bit more in depth about a nucleic acid. Nucleic acids consist of a sugar, which is this orange section down here in this diagram, the phosphate group, which is this turquoise. Is that turquoise? Yeah, this turquoise part. And then nitrogenous base, which is the purple section. And then some differences between DNA and RNA is that the sugar in DNA is deoxyribose versus RNA, which only has ribose. And then the nitrogenous bases in DNA is thymine, whereas RNA swaps thymine with uracil. And we know that DNA is double-stranded, whereas RNA is usually single-stranded, and the two DNA strands are anti-parallel, which again means that they run in opposite directions. All right, so now let's move on to the progress check section. So again, if you would put the answer that you think is correct in the Discord chat, I'll give you guys a couple of minutes to do that. Your question is, which property of water is most responsible for the movement of water from the roots to the leaves of a plant? Well, it looks like most of us are in agreement that it's B. And the correct answer is B. So if you missed this question or had some doubts about the correct answer, down here, after when I reveal the correct answer, you'll also see below all the answer choices, there is a topic followed by its name. That is the topic that this question covers. There might be multiple. Sometimes there's only one, as in this case. So if you did miss this question or just a little bit confused, I would recommend that you review this topic specifically. All right, now here's another one. Which of the following macromolecules is correctly paired with what it does? Looks like we're all in agreement that it is C. And that's correct. It is indeed C. So the topic that this question covers is 1.5. And then that's pretty much all there is. The most important stuff about Unit 1, which just covers the basics, so shouldn't be too difficult. And then Unit 2 is regarding cell structure and function. You can see that it covers these 11 topics. It's about 10 to 13% of your exam. All right, first let's cover plasma membranes. We know that the plasma membrane is a phospholipid bilayer and it has a hydrophobic middle. What a bilayer means essentially is that there are two layers to the membrane, which we can see down here in the diagram. We can also describe the plasma membrane as being a liquid mosaic, and that just means all the proteins, lipids, steroids, and things embedded in the membrane are free to move around. They're not frozen in place or anything like that. And the plasma membrane is selectively permeable. So small nonpolar molecules can pass through the membrane freely. Larger molecules and polar molecules, they're going to require the aid of transport proteins and channels. There is one exception, which is a polar uncharged molecule, such as water. It can pass through the membrane in small amounts, but if we want to pass any significant amount of these molecules through the membrane, we're going to have to use transport proteins or active transport. And then the selective permeability of the membrane creates concentration gradients, which is important for Unit 3, cellular energetics. Specifically, photosynthesis requires a concentration gradient of protons, and so does cellular respiration. We'll see a bit more of this when we move on to Unit 3. And then endocytosis and exocytosis are two terms that you should know for the exam, and if you've forgotten what they mean, they just describe how a cell can take in or spit out anything that it needs or doesn't need, and it does this using vesicles. And water moves from areas of low solute concentration or high water potential to areas of high solute concentration or low water potential. And you can think of this as if you've ever seen one of those like cooking YouTube channels where they teach you how to make a steak. They always say, we're going to dry brine the steak. This will help draw out some of the moisture. That's water flowing from low solute concentration inside the steak to outside of the steak where there's high solute concentration, which was done by rubbing it with salt. And there are three main terms that we can use to describe these situations, where hypotonic means that the surroundings have a lower solute concentration, hypertonic means that the surroundings have a higher solute concentration, and isotonic means that the surroundings and cell have equal solute concentrations. Animal cells prefer isotonic surroundings, whereas plant cells actually prefer hypotonic ones. And this is because that, as we can see in this diagram here on the very right, hypotonic plant conditions help a plant cell maintain its rigidity, whereas animal cells don't have the cell wall to help them maintain that rigidity, which allows us to move around, but also means that if our cells become hypotonic, they can potentially burst. As we can see in the hypotonic solution of red blood cells, there's one that has actually burst. All right, cell compartmentalization. The inside of eukaryotic cells is separated by membranes into compartments. Each compartment has conditions that favor specific reactions to reduce competition. This is kind of like a house where the membranes are the walls of your house, and then each compartment is its own dedicated room. And membrane-bound organelles, which are the mitochondria and the chloroplasts, scientists think that they were once free-living prokaryotic organisms that evolved into the present-day organelles through endosymbiosis. All right, so that was Unit 2, and now we're going to do a quick progress check. Which organelle is primarily responsible for the synthesis of proteins that will be exported from the cell? Max, endosymbiosis is basically, so if you know what symbiosis is, endosymbiosis is basically where these prokaryotic organisms, they decided that there would be both themselves and the eukaryotic cells would be mutually benefited if the eukaryotic cell absorbed the prokaryotes, where the eukaryotic cell would provide protection for the prokaryote, and in return the prokaryote evolved into mitochondria and chloroplasts to produce ATP through cellular respiration and photosynthesis. Alright, looks like most of us are in agreement that it is C. A couple of us chose B and one person chose D. So let's find out the right answer. The answer is indeed C. If you missed this question, review 2.1 and 2.2. It's not the smooth endoplasmic reticulum. No one chose that. Ribosomes in the cytosol. Ribosomes do not produce proteins. If you will review the cell cycle and RNA, DNA synthesis, you'll see that ribosomes, they read messenger RNA and they create proteins that are, they create proteins that are for use inside the cell. The key point here is that the question said proteins that will be exported from the cell, which is the job of the rough endoplasmic reticulum. And the Golgi apparatus acts as an inner cell highway system that transports things from one part of the cell to the other, and also packages different components for use in the cell. Yeah, sorry, I misspoke. Ribosomes do synthesize proteins. I think I corrected myself, but if I didn't, I'm going to make it explicitly clear here. I misspoke. Ribosomes do synthesize proteins. But the key point of this question is that it said the synthesis of proteins that will be exported from the cell. Where, yeah, greens exported here means that they will be taken from inside the cell and passed through the plasma membrane to outside the cell, other parts of the body. Super Dancer 16, the Golgi apparatus, you can think of it as like a packaging center and distribution warehouse where things that other parts of the cell needs will be packaged inside the Golgi apparatus and then distributed to parts of the cell. Your next question is, which of the following statements about the structure of the phospholipid bilayer is true? Alright, looks like most of us chose A. A couple of us chose B, which is just the reverse of A. So let's see which one is correct. The answer is A. The hydrophobic tails of phospholipids face inwards, whereas the hydrophilic heads face outward. And if you really think about it, this kind of makes sense, because if the heads are hydrophilic, then naturally you want to face outward, where we can find all the water in our bodies. So really, to answer this question correctly, really read and know the difference between A and B. I think that might have been where some people got tripped up. But if you did mess up on this question for any other reason, you can find the answer to this in topics 2.4 and 2.5. An example of endocytosis. Yeah, sure. So like, for example, a white blood cell. We know that white blood cells are part of the immune system when we get infected by bacteria. We know that white blood cells come and eat the bacteria to kill them. But since a bacterium in relation to a white blood cell, it's pretty big from the perspective of a white blood cell. So the white blood cell can't simply pass the bacterium through its cell membrane. It has to completely envelop the bacterium, and it does this using endocytosis. So endocytosis is where a cell takes in materials by basically wrapping itself around them. And exocytosis is the opposite, where a cell gets rid of waste material by making a vesicle around that and expelling it from itself. I see several people typing, so I'm going to give them a second before I move on. Yeah, okay, so however you want to pronounce that, pinocytosis and phagocytosis, however you want to pronounce those, everyone is right. They're pretty much just the cell drinking and eating. Phagocytosis is where the cell basically eats things. You can remember this by, well, I guess the only way you could remember this reliably is thinking of a phage, spelled Phage. That means to eat. So you can see phagocytosis is the cell eating things, whereas pinocytosis is basically the cell drinking. The cell intakes extracellular fluid and smaller molecules through small vesicles. And then, on the other hand, phagocytosis is the ingestion of larger particles like bacteria, which white blood cells do through, well, larger vesicles. I don't remember exactly if my biology class covered it, but I'm pretty sure the AP exam did not go over phagocytosis and such, but it definitely did go over endo and exocytosis. All right. Now let's talk about Unit 3, Cellular Energetics. Now, a couple of days ago, I did do a lecture on specifically targeting Unit 3, but due to some technical errors, that didn't start until 8, so my apologies for my end. The recording is available to view, and as such, because of that lecture that I did specifically targeting this unit, I can't go into too much detail here. So if you do have other questions that this overview did not answer, I would recommend that you go and watch the recording there. Where can we get the recording? I will go get that link and put it in here. Be right back. Okay, so I've just posted a document in the lecture stage chat, which links to our lecture schedule, and you can see in the columns both our lecture slides and our lecture recordings. All right, so cellular energetics. It only has 7 topics, but for such a short unit, it takes up a lot of the exam, 12 to 16%. So first, we learned about enzymes. Just a quick overview. Enzymes are proteins that catalyze a reaction. They have an active site where the reaction takes place, and this active site has to have the correct shape and charge for the reaction that it catalyzes. And if you'll recall from Unit 1, which we just went over, proteins are complex polymers that consist of amino acids that fold into a specific shape. And it's this shape of the protein that determines its function. And proteins can also denature, which therefore means that enzymes can also denature. Some enzymes can reform when they are placed back into the correct conditions. And on the bottom diagram here, we have competitive inhibition versus non-competitive inhibition, where competitive inhibition means the inhibitor comes and occupies the active site. Non-competitive inhibition means the inhibitor occupies an allosteric site and changes the shape of the active site, thereby also preventing the reaction from happening because the reactants cannot fit into the active site. And then an overview of photosynthesis. Obviously, this happens only in plant cells because only plant cells have the correct organelles to do so. We see first that light powers the light reactions, which takes in water and light as reactants, generates oxygen, the molecule ATP, and the electron carrier NADPH. And these two molecules power the Calvin cycle, which takes in carbon dioxide and produces sugar in the form of glucose, which is the plant's main food source. So light-dependent cycle, I'm only going to go over this once because of time constraints and the fact that we also still have like 60% of the course to go through. So light energy from the sun strikes photosystem 2, which bounces the light energy back and forth like a ping pong ball between a bunch of chlorophyll atoms until it is channeled down here into a centralized pair of chlorophyll molecules. And this centralized pair is excited by the light energy and releases an electron. This electron travels through the membranes of the chloroplast, and this energy from the electron helps to create a hydrogen-proton gradient, which we can see there's a lot more protons down here in the interior of the organelle versus outside in the stroma. Now, this electron travels to photosystem 1, and the energy from that electron combined from light energy from the sun does pretty much the exact same thing. It is channeled down into a centralized pair of chlorophyll molecules, and that pair of chlorophyll molecules, once again being excited, releases an electron, which travels down through another electron transport chain. But the goal of this chain here is that the electron ultimately turns NADP plus back into its energized electron carrying form, NADPH. And then after we've created this hydrogen proton gradient, they're going to all rush back out to equalize the concentration of hydrogen protons on either side of this membrane. And they mainly travel out through this protein, the channel protein ATP synthase, which harnesses the energy from the hydrogen protons to pin a phosphate group onto ADP to turn it back into ATP. And over here, we can see where the water comes into play. We need to replenish the electron that traveled from photosystem 2 all the way down through the other photosystem into NADPH. And well, where do we get that other electron from? We can do that by splitting a water molecule down into oxygen and hydrogen, which releases one electron that we can then take back into photosystem 2 to replenish the electron that we lost. And then taking the products of the light-dependent cycle, NADPH and ATP, we can then go through the Calvin cycle. We're going to take 3 carbon dioxide molecules, turn them into 6 3-PGA molecules through a process called carbon fixation, and then through another process called reduction, which takes the energy from ATP and NADPH. We're going to get 6 G3P molecules, and of those 6, only one of them will actually go to make glucose. 5 of them are recycled in a process called regeneration, which also takes energy from ATP. And these G3P molecules are regenerated back into the starting molecule, RUBP. And we see that glucose down here, our product, is ultimately what becomes the plant's food source. And then for the scope of the bio exam, you won't need to know all these different chemicals and molecules, compounds, all that. Mainly what you do need to know is that step 1, we take CO2 in a step called carbon fixation, combine it to create 3-PGA, and then those 3-PGA molecules turn into G3P, one of which makes glucose, and the other 5 turn back into the starting product, the starting reactants. And then the animal cell counterpart to photosynthesis is cellular respiration. And this is what we do, what every other animal on Earth does in the mitochondria. So it starts via glycolysis, which is where we split one glucose molecule into 2 pyruvic acid molecules. This generates 2 ATP. And then our pyruvic acid goes into the Krebs cycle, which transforms the chemical energy to power the electron transport system, which is what creates the bulk of our ATP. 36 molecules you'll see here. So one cycle of cellular respiration produces 38 ATP molecules. Now this is a very best case estimate. So the absolute maximum number of ATP molecules you can get out of one cycle is 38. A more realistic number is usually 30 to 32 because of the fact that we need ATP to power most of these processes, things like that. So here we've got a more sort of detailed diagram. We see that in glycolysis, we take glucose, we split into 2 pyruvate molecules. This generates ATP and the electron carrier NADH. And then step 2, our pyruvate oxidation. We take pyruvate, turn it into a molecule called acetyl coenzyme A, or acetyl CoA for short. This produces carbon dioxide and some more NADH. Then our acetyl CoA goes to the citric acid cycle, which is also a long, complex process similar to the Calvin cycle here. But you don't need to know all the different processes and molecules involved. You just need to know that acetyl CoA goes in, produces ATP, carbon dioxide, NADH, and then this other electron carrier, FADH2. And then finally, all these electron carriers and such, they go into oxidative phosphorylation, which we're going to look at here. This is very similar to photosynthesis in that we're trying to create a proton gradient that can then turn our ADP back into ATP. So first, we see that our products from the citric acid cycle, we have NADH and FADH2. So our NADH is going to be attracted to complex 1 embedded in the membrane of the mitochondria. So it's going to donate an electron. It's going to split back into NAD plus and a hydrogen proton. And then our other electron carrier, FADH2, is going to do the same thing, except it's going to donate its electron to complex 2. Now, all these electrons keep traveling along the electron transport chain through these other complexes, complex 3, site C, and then ultimately complex 4. And all the while, it's helping generate this proton gradient. And then at the end of your electron transport chain, it's going to pop back out. And then we're going to be able to take that electron, combine it with oxygen and hydrogen, and produce water. So basically the opposite of what we did in photosynthesis. And then notice now how we've got a whole bunch of protons in the inner membrane space of the mitochondria. So now they're going to all come rushing through ATP synthase, just as they did in photosynthesis. And then ATP synthase is going to harness the energy from the proton flow to be able to tack on a phosphate group onto ADP to once again generate ATP. Now, you'll notice that the name of this process is oxidative phosphorylation. That tells you that this process, alongside the Krebs cycle, all require oxygen to run. But what happens if we don't have any oxygen? For instance, you're running a marathon, and eventually over time, your muscles use up oxygen faster than you can replenish it. Well, it's not the end of the world yet. We can resort to a process called fermentation, which is what happens when we need energy, but we don't have the oxygen to go through oxidative phosphorylation. So we'll see that the first step in fermentation is glycolysis. That stays the same, where we're taking one glucose molecule, splitting it into 2 pyruvate molecules, getting 2 ATP molecules out of it and 2 NADH electron carriers. But then since we're missing oxygen, we can't run the oxidative phosphorylation process, which generates the bulk of the ATP. So then we take our 2 pyruvate molecules and turn them into lactate, which is lactic acid, in a process called NAD plus regeneration. And this is where we can regenerate, where the NADH electron carrier donates its electron, turns back into NAD plus to go back into the glycolysis step. But lactic acid, is considered a waste product by our body. So lactate is just going to build up over time in your cells, and eventually we're just going to have to stop exercising, period, to get rid of all of that. But the main point here is that when we run out of oxygen to power the oxidative phosphorylation part of cellular respiration, we can resort to fermentation, which is a whole lot less efficient. As you can see, a net of 2 ATP versus a net of 38 ATP. But this is what enables us to keep going after we've exhausted oxygen reserves. And you'll see that in prokaryotes, which do not have mitochondria, their only way of making ATP is through fermentation. So that was a very long explanation of a lot of stuff at once. So if anyone has questions or wants me to go back and look through something again, now is the time to ask. If not, here is your progress check question. Which of the following statements correctly describes the role of ATP in cellular processes? You don't necessarily have to memorize the amount of stuff going in and out to answer your question, Tater. But if you look at my most recent message in the chat, you should know the names of the electron carriers as well as how much ATP is produced. Those are the most important things to know. Alien, to answer your question, the electron carriers in photosynthesis are NADPH. This red, what is represented by this red oval in this diagram, NADPH is our electron carrier for photosynthesis. Whereas in cellular respiration, our electron carriers are now NADH and FADH2. Now you should also remember that NADPH, after donating its electron, turns into NADP+, whereas NADH turns into NAD+, and FADH2 turns into FAD. NAD plus, who asked that question? Yeah, okay. NAD plus is basically let me find that diagram. NAD plus is the form of NADPH after it has donated its electron. And its main job is to go back into the light-dependent cycle, pick up another electron, turn back into NADPH, so that it can then help power the Calvin cycle by donating its electron again. So it constantly alternates between the forms NADPH and NADP+, depending on whether it has that electron available to power the Calvin cycle. Alright, hold on. Let me see what the answer of our poll is. So most people are agreeing that the answer to this is B, and that is correct. The topic that this question covered was 3.4, cellular energy. So if you had questions about it, I recommend that you review that topic. And then if you do have any other questions, once again, do check out the linked spreadsheet that I provided earlier, which you can find the recording for the Unit 3 lecture that I did a couple of days ago, which goes more in depth into what we just covered. So now, for the sake of time, we need to move on to Unit 4, Communication and Cell Cycle. So this is about 10 to 15% of your exam, which for such a short unit is also a pretty significant weighting. So, cell communication. Cells can communicate with other cells that they are touching, but oftentimes we want to communicate with cells on the other end of our body, for instance. To communicate over short distances, our cells can emit chemical messengers called ligands. And when a cell receives a ligand, a receptor is going to receive the ligand in the target cell, and the receptor starts to change shape. And when it does so, it releases other messenger molecules, which amplify the signal and start a signaling cascade inside the target cell, where other receptors receive these released messengers and release their own messengers, so on and so forth, until the desired change in the target cell happens. Long distance communication, so from your head down to your leg or something like that, these rely on hormones in the endocrine system. And finally, mutations at any point in the transduction pathway can alter the subsequent transduction of the signal, resulting in an incorrect response. So you can think of this like when you're doing a long math problem, where to complete the next step, you need the answer from the previous step. So if you make one small mistake, like accidentally write minus instead of plus, you're going to end up with the wrong answer for one step, which is then going to lead into the wrong answer for the next step, so on and so forth, until you get to the final answer, which is wrong because you missed a plus and put minus instead. Alright, the cell cycle is a highly regulated process for the growth and reproduction of cells. There are 3 stages of interphase, followed by mitosis, which is where the cell divides, and cytokinesis, when the cell's cytoplasm splits into 2 to finally form 2 distinct cells. And then during interphase, if the cell determines it does not have sufficient resources or room to divide, it can enter the G0 dormant phase and can be pulled back into the cycle once there is enough resources or room. And the interactions between cyclins and cyclin-dependent kinases control the cell cycle. Disruptions to the cycle or disruptions to the cell cycle, or if a cell is found to be unfit, will result in apoptosis, which is pre-programmed cell death. However, in the very, very minuscule chance that apoptosis doesn't work, or if a cell somehow escapes detection, then it is likely to keep mutating until it turns into a cancerous cell. And there are three main checkpoints that check that the cell is actually viable in the cell cycle. At the end of the G1 stage in interphase, one checkpoint verifies all the DNA carried by the cell is viable, there are no errors in it or whatever like that. And at the end of the G2 stage, another checkpoint verifies that chromosome duplication was successful. So at the G2 stage, we're just about ready to begin mitosis. All our chromosomes have been duplicated in theory, and this is what the G2 checkpoint checks for. And then during mitosis, a third checkpoint verifies that each centromere is attached to a spindle fiber, so that each daughter cell will get the correct amount of genetic material. Here we can see the entire cell cycle, where our interphase includes the phases G1, S, and G2, whereas finally during cell division we have mitosis, our 4 stages of mitosis, and then finally cytokinesis. You can see on the top left-hand corner G0, which represents the dormant stage. The cell that goes into the phase G0 is not dead. It's just being commanded to not divide until it either has enough room or has enough resources to complete mitosis. So a bit more about mitosis. You should know that it is where one parent cell splits into 2 genetically identical daughter cells. Down at the bottom, we can see our diagram of mitosis, where it starts with prophase, proceeds through metaphase, anaphase, telophase, and then finally cytokinesis splits the 2 cells into 2 distinct cells. Before prophase, each chromosome is duplicated and DNA coils into the chromosomes. And then in prophase, centrioles, which are organelles in the cells, move to opposite ends of the cell. The nuclear envelope containing the genetic material dissolves. In metaphase, the chromosomes all line up in the center of the cell. The centromeres develop spindle fibers that attach to each chromosome, and the organelles disappear. In anaphase, the spindle fibers are going to pull half of each duplicated chromosome to opposite ends of the cell. And then in telophase, the nuclei reform, spindle fibers break down, the chromosomes unwind back into the loosely coiled genetic material in the nucleus of a normal cell, and the organelles reform. And then finally, as we've said, cytokinesis, the cytoplasm splits and divides the cell into 2 distinct cells. So now is the progress check, where which of the following events occurs during the G1 phase of the cell cycle. And now I'm also going to look in the Discord chat and see if anyone has questions regarding Unit 4. So Max's question is the S phase both DNA and chromosome replication, or is it just DNA? So during the S phase, let's go back to our thing. During the S phase, it's where a chromosome is just coiled up DNA. So during our S phase, the main job is to duplicate the DNA. Whereas the actual chromosome replication part doesn't happen until actual mitosis. All right, based on our poll, I can see that most of us chose D, so let's find out. It is D, so this question had to do with 4.6, the cell cycle. So if you missed this question or I guess had questions about the question, check out 4.6. All right, your other question is, which of the following is a correct statement about signal transduction pathways in cells? All right, so looks like we've got most people think it's C. A couple people think it's A, so let's find out. The answer was indeed C. So let's take a look at A and figure out why it's not correct. So A says the signal molecule binds to a receptor on a cell surface, and the response is immediately carried out in the cytoplasm without any intermediate steps. So this answer seems correct at first, but it is not correct because it forgets one point in signal transduction, which is the third bullet point here. It omits a signaling cascade. So one receptor receiving a ligand and changing shape most of the time is not enough to trigger the response. Because if you think of it, a cell is orders of magnitude larger than a signaling molecule or a receptor. So receptors rely on a cascade of signals to create a large enough signal for the cell to actually start doing things. So to answer your question, first we have direct cell-to-cell communication. So basically one cell communicates directly with its neighboring cells that touch them. And then this is also going to answer your question, the guy with 7 AP tests this year. So on the most local scale, cells directly want to communicate with other cells that are touching them. They can just send chemical messengers directly across their cell membranes to talk to each other. This is the most local in scale, so we really can't go anywhere beyond like a couple cells away. But then if we want to target cells that are a little bit farther away, we can rely on paracrine signaling, where we can let our signal molecules diffuse over short distances to affect nearby cells. And then finally, endocrine signaling is where, let's say, our brain wants to tell, what's a good organ? Our brain wants to tell the large intestine to do something. To do this, we need to rely on the endocrine system, which transmits hormones through the bloodstream to the target cell. And a hormone is basically just a signal molecule. So neurons are different. Used in cellular communication, we're interested in chemical signal molecules, such as adrenaline, whereas where's your question? Whereas neurons transmit electrical signals, not chemical signals. All right. And the unit 5 is heredity. Covers these 6 topics and is weighted about 10% on your AP exam. So the first first major topic in this unit is meiosis. Different from mitosis, meiosis produces haploid gametes, otherwise known as your sex cells, in sexually reproducing diploid organisms. And the resulting daughter cells only have half of the chromosomes of the parent cell. And what this means for gametes is that they need to fuse with another gamete to have the correct number of chromosomes to create a new organism. And we can see that a complete cycle of meiosis actually has two different steps. The first is meiosis 1, and then it does it again to create 4 daughter cells from one parent cell, whereas in mitosis, it only creates 2. So if you did want to look over the diagram posted, the slides are available in the lecture chat. And then after that, after meiosis comes Mendelian genetics, which stem from the research of Gregor Mendel, the monk with the pea plants. And he figured out that genes come in different versions. He called them alleles, and the dominant allele will always hide a recessive one, pretty much always. And then he came up with 2 laws that describe what he found with alleles. And the law of segregation states that each gamete will only receive 1 copy of an allele. And his other one, the law of independent assortment, states that the allele a gamete gets for one gene does not influence which one it gets for another gene. So, for instance, the allele that a gamete gets for, eye color is not going to affect the allele that it gets for hair color. And then we can also use Punnett squares to determine the genotype and therefore the phenotype, which is the physical expression of the genotype of the offspring of 2 organisms, provided that we know the parent's genotype. The rules of probability apply to the analysis of the passing of these single gene traits, and then we can predict the pattern of inheritance, whether that's monohybrid, dihybrid, sex-linked, or genetically linked genes. We can predict these patterns from data that we collected. However, later scientists went on to discover that the inheritance of many traits do not follow Mendel's laws, and these are called non-Mendelian genetics. So, genes that are close to each other on the same chromosome, they appear to be genetically linked. And the probability that genetically linked genes segregate together can be used to calculate the distance between them. So basically what this is saying is that Mendel previously thought that the law of independent assortment, the allele that a gamete gets for one gene, does not influence which one it gets for another gene. But later on, we found out that genes that are close to each other on the same chromosome have a greater likelihood of being passed together. And we also figured out that many traits are the product of multiple genes or physiological processes, so they do not segregate in Mendelian patterns. Otherwise, in simple terms, there's no clear difference between phenotypes. And then finally, other traits result from non-nuclear inheritance. And the 3 main examples are listed. The chloroplast and mitochondria, which are the energy-producing organelles, are randomly assigned to the gametes and daughter cells. So traits inherited from mitochondrial or chloroplast DNA are not going to follow Mendel's rules. In animals, the mitochondria are transmitted by egg cells. So all of your mitochondria you got from your mother. Thank your mom for that. So traits determined by mitochondrial DNA must be maternally inherited. And then also in plants, both mitochondria and chloroplasts are transmitted in the ovule, which is basically the plant's version of a female gamete. So, and then you should have learned about the 5 patterns of chromosomal inheritance, which are summarized here. Autosomal dominant and recessive. Autosomal dominant, the trait appears in every generation, and each affected individual is going to have at least one affected parent. For autosomal recessive, the trait could skip generations, and it's usually appearing only when an individual receives 2 recessive alleles, which makes sense because if a dominant allele is going to mask a recessive one, then we're going to need 2 recessive alleles for the trait to show up. And then X-linked dominant, the trait doesn't skip generations. An affected father only passes it to his daughters, but an affected mother can pass it to both her sons and her daughters. We also have X-linked recessive, which states that more males are affected, and the trait can skip generations. Affected males don't pass the trait to their sons, but they can produce carrier daughters, and a carrier in this context means that the daughter carries one dominant allele and one recessive one. So the daughter does not display the phenotype, but she carries the genetic material for the affected phenotype. And then finally is Y-linked, where only males are affected and fathers pass the trait on to sons. And the part where only males are affected is because these traits are only found on the Y chromosome, which only males possess. If you'll remember, to create a female offspring, you need two X chromosomes, whereas a male requires one X and one Y. Alright, so now is your progress check. Which of the following is true about Mendel's law of independent assortment? Alright, now that I've answered a couple questions, our poll states that 80% of you think it's C, 15% think it's D, and 1% thinks it's A. The correct answer was C. To those people that didn't get the correct answer, I recommend that you review 5.3 Mendelian Genetics. So A looks to be true at first glance, but it says alleles for different traits are inherited together. This implies that they will always be inherited together, which is not what we know to be true based on modern science. And the question also asks about Mendel's law of independent assortment. So we go back to Mendel's law. We see that it states the allele a gamete gets for one gene does not influence which one it gets for another gene. So A isn't addressing what the question is asking, making that one incorrect. And then D explains how homologous chromosomes are paired up during fertilization. This is a mitosis and meiosis answer, not about Mendel's law of independent assortment. And then your second progress check question is, in a dihybrid cross between two heterozygous individuals and their genotype is given, what is the probability of producing an offspring with the genotype of big Y, big Y, big R, big R? Let's try to keep the lecture chat limited to AP biology concepts out of respect for everyone that showed up today. So looking at the poll, looks like most of you selected A. A little bit, a couple of you guys put B. So let's see the correct answer. The correct answer is 1 out of 16, otherwise known as A. If you missed this one, study 5.3 Mendelian genetics, also Punnett squares, which is how you can get the answer here. And then let's move on to Unit 6, which is gene expression regulation. This one is also a bit memorization heavy, and it's 12 to 16% of the exam, so time to focus. So first is DNA replication. DNA is synthesized from the 5' end, and one strand is the template for a new complementary strand. And there are 4 main enzymes here that we are concerned about in DNA replication, namely helicase, topoisomerase, DNA polymerase, and ligase. So helicase comes in first and unwinds the DNA helix and separates the strands into 2 distinct strands. Topoisomerase goes ahead of helicase and relaxes supercoiled DNA so helicase can properly unwind it. And then DNA polymerase uses RNA primers to synthesize DNA. And because we can only synthesize DNA from the because we can only synthesize DNA in one direction, DNA polymerase is going to synthesize DNA continuously on the leading strand, and then in segments on the lagging strand. And then we have to somehow connect these fragments together on the lagging strand, which is where ligase comes in. Ligase is what joins the fragments together on the lagging strand. I'm going to see down here a quick little diagram of what's going on here. You see the green hoop of topoisomerase is relaxing the DNA to let helicase, the blue triangle, come in and unzip it. And then DNA polymerase, the yellow orangish rectangles, are coming in synthesizing DNA, and then finally DNA ligase is joining the fragments together on the lagging strand. And notice that these fragments on the lagging strand, they have a name called Okazaki fragments. I don't think that's too important for the exam, what those fragments are called, but it's good to know in case you do get tested on those. And then is transcription and RNA processing. So there's a bunch of different types of RNA. First is mRNA, stands for messenger RNA. This RNA is going to be what carries information from nuclear DNA, which is what's DNA just stored in the nucleus of the cells, to the ribosomes. And then tRNA, transfer RNA, is what's going to bind to specific amino acids and have the anticodon sequences that pair with mRNA to synthesize proteins. And then rRNA is what actually forms the ribosomes. So in the ribosome is where protein synthesis happens. And basically mRNA brings the instructions, tells the instructions to tRNA, which goes out and finds the specific amino acid to build the protein. And this all happens in the ribosome, which is formed by rRNA. So there's 3 main types of RNA here that we're concerned with. And to summarize what just happened is genetic information flows from DNA to mRNA through a sequence of amino acids. The process by which RNA polymerase create a strand of RNA from a template strand of DNA is called transcription, which basically it starts when the mRNA copies what the DNA says, brings it into the ribosome for tRNA to bring it the specific amino acids to create the protein. In eukaryotic cells, the mRNA transcript gets a poly A tail and GTP cap, which are both just a bunch of different molecules. Introns are removed and exons are spliced. And this is how genes are actually expressed. And alternative splicing is where different versions of mRNA are created based on which introns are deleted and which exons are kept. And translation is different from transcription in that translation is the process by which mRNA is read by rRNA to create polypeptides. In prokaryotic cells, so like your bacteria, translation and transcription occur together, but not in eukaryotic cells. And then during translation, nucleotides from the mRNA are read in sets of 3 called codons, where each codon represents a specific amino acid. And if you're ever asked on the AP exam to translate a sequence of mRNA, you're going to be provided the genetic code chart. And almost all organisms use the exact same genetic code for its amino acids, which is also a sign of a common ancestor. tRNA brings the correct amino acid to the end of the polypeptide chain, and this continues until the stop codon is reached, upon which the ribosome releases the polypeptide chain. And then in retroviruses, the genetic information is instead coded in RNA, and an enzyme called viral transcriptase inserts its genetic information copies it into DNA, which is then inserted into the host genome. So the virus basically uploads its own information into the host's genetic information. And then regarding the genetic code chart, there are only 20 amino acids, but we can create a lot more than 20 distinct combinations using 3 individual letters for a codon. And so a bunch of codons will code for the exact same amino acid. So sometimes you can have a little typo that results in a changed codon, but this codon is going to code for the same amino acid. So at the end, nothing happens. The polypeptide chain forms normally. It folds into the normal protein. Everything is well. All is fine. And as we've said before, for regulation of gene expression, regulatory sequences of DNA interact with regulatory proteins to control transcription. The phenotype of the organism is determined by the combination of genes expressed and, at the same time, to what degree they are expressed. Operons are groups of genes that are transcribed in a single mRNA molecule, and groups of genes can be influenced by the same transcription factors to coordinate regulation. I apologize if I'm starting to go a little fast, but I am limited to a 2-hour lecture window. We are only on Unit 6 out of 8. And then last in this unit is mutation, where the normal functioning of the genes and gene products results in normal functioning of an organism. So basically, presumably, so basically what this is saying is that the result of the normal functioning of a gene is the normal functioning of an organism. So like a healthy plant that doesn't randomly grow apples when it's supposed to grow oranges, which I don't know how that would even happen, but just an example. Disruptions in genes and gene products can create new phenotypes, and DNA mutations can be positive, negative, or neutral. Now, most of the time, whenever DNA replication errors happen a lot more often than you think. It's just that they're usually caught in time by these checkpoints in the cell cycle, which you'll remember from a couple units ago. But sometimes DNA mutations are let through because nothing catches them. And then most of the time, most of these mutations that are accidentally let through ultimately result in nothing. It's going to take the mutation of a lot of stuff at once, or the mutation of one very specific, very important gene to actually result in a physically detectable mutation, such as, I don't even know what it would be. I don't know, like 6 fingers. Mutations are the primary source of genetic variation. And finally, changes in genotype can produce phenotypes that are subject to natural selection. And natural selection is Unit 7, which we are about to get to shortly. So your progress check question is, which of the following processes occurs in the nucleus of eukaryotic cells and is the first step in gene expression? All right, looks like we have about a 50-50 split between B and C. So the correct answer is C. The correct answer is not B because the question says the first step in gene expression, whereas DNA replication refers to the duplication of DNA in preparation for mitosis, which mitosis is a separate process from gene expression. And the first step in gene expression is transcription, where our messenger RNA copies what the DNA says and then travels to the ribosomes. So further information, definitely review 6.3, transcription and RNA processing. I see this one tripped up a lot of you. Yeah, if this question instead said, what is the first step in replication, then out of these four answers, if this question was instead asking, what is the first step in mitosis, then out of these four answer choices, the correct answer would be DNA replication. So the reason why the answer to this question is C and not B is because the question says, what is the first step in gene expression? And the reason it's not B is because DNA replication is not involved in gene expression explicitly. DNA replication refers to the process where we take the parent cell's DNA, replicate it so that we have two copies of it in preparation for mitosis. Whereas in gene expression, the correct first step is transcription, where messenger RNA, mRNA, copies the DNA and then takes its message to the ribosomes in the cytoplasm of the cell. Your second question is, which of the following is an example of gene regulation in eukaryotic cells? All right, looks like most of us think the answer is A. Let's find out. The answer is A. So in eukaryotic cells, one of the main ways that we can regulate genes is adding the cap and a poly A tail to mRNA. For those of you that missed the question, check out topic 6.5 and 6.6, which are regulation of gene expression, mainly that one, and then also gene expression and cell specialization. So on to Unit 7, which is the longest unit in AP Biology. This is 13 to 20% of the exam, but it's also 13 different topics. So about how much I would expect on the exam for such a long topic. Okay, yeah, looks like they answered Max's question. So first thing in Unit 7 that I thought was important that you talk about was Hardy-Weinberg equilibrium. And the Hardy-Weinberg model, essentially what that does is it helps us predict how common is an allele going to be in a non-evolving population. But what is a non-evolving population? A non-evolving population for the context of Hardy-Weinberg equilibrium is defined as 1, having a large population size, 2, an absence of migration, 3, no net mutation, 4, random mating, and 5, absence of selection, whether that's natural or artificial selection. So if our population meets all these, requirements, then we can apply the two formulas to calculate allele frequencies. p squared plus 2pq plus q squared equals 1, and p plus q equals 1, where p and q are the frequencies of a certain trait in the population. And changing allele frequencies provide evidence for evolution, and small populations are more susceptible to random environmental impacts. Common ancestry. All life is thought to have originated from a single species, and there's a bunch of evidence that argues for this concept, such as pretty much all complex life having mitochondria, a lot of organisms having very much of their DNA in common, things like that. Across all eukaryotes, there exist, as I've said, memory-bound organelles. We also have linear chromosomes, and we also use introns in gene regulation. Many of the fundamental features and processes are conserved across organisms, such as cellular respiration, most importantly. And the appearance of new species can be described using a cladogram, which is this little figure at the bottom of the slide here. So basically, each branch represents a separate species, and then the trait that defines all the subsequent species is listed by these little black tick marks. So at the very left, we have tortoise, and the tortoise is different. It's split off because it doesn't have hair. And then comes the horse, because it isn't carnivorous, and then the wolf, because it doesn't have retractable claws, and then finally the leopard is split off from the house cat, because the leopard cannot purr. And that ties into evolution, which so I think at this point everyone should know what evolution is. It's supported by evidence from many disciplines, not just biology, also things like geology, obviously from fossils, things like that. But the point is, a lot of evidence from a vast variety of scientific disciplines supports the concept of evolution. Vestigial structures are also evidence of evolution and common ancestry. So if you ever looked at a whale skeleton, you'll see that on their pelvis they have these little vestigial bones, which are the remnants of what used to be hind legs. So a long time ago, what was the ancestor of a whale used to be a four-legged land mammal. Well, it may not have been a mammal, but the point is, it had four legs, it walked on land. And so eventually, over millions of years, its front legs turned into the whale's pectoral fins, but it never had use for the back legs, so they just kept, they had no use for the back legs, so eventually they reduced down into these tiny little bone stubs, nubbins, that you can see on a whale's pelvic girdle if you ever look at that. And even today, populations of organisms continue to evolve. We can see this by genomic changes over time, continuous change in the fossil record, and a modern example of this is bacteria becoming resistant to drugs or pesticides. There was an experiment run by Harvard Medical School where they decided to see how long it would take for a population of bacteria to resist 1,000 times the antibiotic that the original strain could, and it only took 11 days for a new population of bacteria to emerge that was 1,000 times more resistant to antibiotic. And this also leads to new and emerging diseases, where old diseases continue to evolve to the point where it's considered as a new one that we have to figure out how to fight. And this brings us to convergent and divergent evolution, where convergent evolution describes the appearance of similar traits due to similar pressures between species. Like birds, bats, and insects all developed wings, but obviously at different times, and their wings are not structurally the same. But the fact that they all evolve wings, they evolve wings due to similar pressures between the species. And then divergent evolution, on the other hand, describes when two populations are geographically separated and evolve differently to suit their habitats. So for this one, think about Charles Darwin and his finches that he found on the Galapagos Islands. They're all finches, but each of them had a distinct beak shape to suit their niche in the environment, whether that was catching insects or crushing seeds. Each one of them, because they were geographically isolated on different islands, eventually evolved to fill a different niche and to prey on different things. So their beaks evolved to give them the best advantage at catching whatever it is they eat for food. And phylogenetic trees show the amount of change over time and show traits that have been lost or gained during evolution. And we construct phylogenetic trees and cladograms from either morphological similarities between species or fossils and DNA similarities. And so at what point do we consider a population to be a new species? Well, the answer to that is when its members no longer breed with members of the original population to produce viable offspring. And a viable offspring is defined as one that will grow up healthily and then grow up to be able to produce offspring of its own. So, for instance, a horse and a donkey are two separate species because while they can breed to produce a mule is sterile. So therefore, a mule is not technically considered a viable offspring. And the speciation rates can become especially rapid during adaptive radiation when new habitats become available. And finally, speciation is what results in the diversity of life that we see on Earth today. We also have extinction, where not a whole lot to cover here that isn't just common knowledge, but the most important things to summarize here are that the amount of diversity in an ecosystem can be determined by both the rate of speciation, which is the rate at which new species evolve, and the rate of extinction. And extinction frees up previously occupied niches that can be exploited by other species. And finally, you have variations in populations. We can see in this picture on the right here, all of these shells are from the same species, but no two shells look exactly alike. This is a great example of just some of the genetic variation that can happen even within a population of the same species. So genetically diverse populations are more likely to contain individuals who can withstand extreme environmental pressures. So a more genetically diverse population is more resilient. Although beneficial for adaptation in one environment, can also be harmful in another. So that is to say, context is very important in biology. So your progress check question is, which of the following is the best definition of natural selection? Looks like 100% of you said A, which is the correct answer. If you had questions about this question, this one was written to cover 7.2 natural selection and 7.4 population genetics, so check those out if you were confused. And then your next question is, which of the following is the best explanation for allopatric speciation, where allopatric speciation describes the emergence of new species due to, if I explained it would give away the answer. So I'll tell you what allopatric speciation means after you guys answer this question. Looks like a lot of us picked A. Let's find out if that's right. Whoops. The correct answer is A. So allopatric speciation is when a new species emerges due to geographic isolation from its original population. So maybe let's consider a population of birds, and one day a big storm blows a small population of these birds offshore maybe a couple hundred miles away, onto an island. And over a long time, the original population of birds evolves differently than the population that was blown onto the island because they experience different environmental pressures and also maybe fill different niches in the environment. So over many generations, they eventually evolve enough to be considered two separate species. And so this is what allopatric speciation is, which is reflected in answer choice A, when populations become reproductively isolated due to physical barriers leading to genetic divergence. All right, Unit 8 is ecology. It's 7 topics. It consists of 10 to 15% of the exam. This is also the last unit, so let's get started. Final sprint. So organisms respond to changes in their environment through behavioral and physiological mechanisms. And organisms also exchange information with one another, which changes their behavior. And individuals can act on this information and communicate it to others. Responses to information and its communication are vital to natural selection and evolution. So what does it mean by organisms exchange information with one another? There's a whole lot of ways that organisms exchange information with one another. The most obvious one is the spoken language, which we are doing right now. I'm presenting this information to you. But also consider, for example, a beehive. When a bee goes out and identifies a patch of flowers, it comes back and it tells the hive where these flowers are by dancing. And then so these bees will be like, hey, there's a bunch of flowers over there. And so they're going to go out, look for these flowers, and bring back their nectar to turn into honey. So this is how organisms can change their behavior by exchanging information with one another. Organisms use energy to maintain organization, otherwise known as homeostasis, grow, and reproduce. Any changes in the availability of energy can result in changes in a population's size and disrupt the ecosystem. And regarding a food web or perhaps trophic cascades, there are two main groups that we're concerned about. An autotroph, which is like a plant, anything that can capture energy from physical or chemical sources in the environment, which is the fancy way of saying can make its own food. And then heterotrophs, which the scientific method of describing them say they capture energy present in carbon compounds produced by other organisms. In normal speak, this just means heterotrophs eat autotrophs to gain energy. Many adaptations in organisms are related to obtaining and using energy and matter. And the structure of a community is measured and described in terms of both species composition and species diversity. So basically, how many species are there? Is this area diverse? Communities change over time depending on interactions between populations, and relationships among interacting populations can be, relationships among interacting populations can be characterized as positive or negative and can be modeled. And these include competition, so competing for food or for resources, predation, trophic cascades, niche partitioning, and symbiosis, which such as parasitism or commensalism or mutualism. And these relationships drive population dynamics. So basically how one population is going to interact with another. Biodiversity. Ecosystems with fewer components and less diversity are less resilient to changes in the environment, kind of like how a population with less genetic diversity is more vulnerable to sudden pressure from their environment. Keystone species producers and essential abiotic/biotic factors contribute to maintaining diversity. What is a keystone species? A keystone species is a species whose effect on the population is disproportionate relative to their abundance. So they have a very large effect on their environment when compared to how many of them there are. And if you remove the keystone species, the ecosystem just collapses. And probably the most famous example is the Yellowstone wolves, which we'll examine in the progress check question. And finally, disruptions to ecosystems. Just to review, an adaptation is a genetic variant favored by natural or artificial selection and provides an advantage to an organism in its environment. But an invasive species will exploit a new niche free of predators, and it will outcompete native organisms for resources, and such as bottom here, the lionfish, which is native to, I believe, the oceans of Southeast Asia, but it's been introduced pretty much globally now, and since it has venom, it's also got giant spines that make it very difficult to eat by native predators, and they also reproduce at obscene rates. So pretty much anywhere where the lionfish has been introduced, it has been overrun by them, and they'll eat pretty much anything that'll fit in their mouths. And then finally, the distribution of local and global ecosystems changes over time, and human impact accelerates change at local and global levels. So tying this back to the lionfish example, how long do you think it would have taken for a lionfish to swim from Southeast Asia all the way over to Florida? Probably never. But thanks to humans, some lionfish hitched a ride over on a boat, and now people are literally paying fishers to go out and target lionfish. So your first progress check question is, which of the following is example of a density dependent factor that affects population size? All right. Looks like 100% of you selected B as the answer, which is the correct answer. So this question was written to cover 8.4 and 8.5. So if you wanted to know more or had any confusions, check out those topics. And then, as I promised, the progress check about wolves. When wolves were eliminated from Yellowstone National Park in the early 20th century, elk and deer populations grew out of control, and subsequent overgrazing destroyed much of the park's habitats. Yellowstone's wolves are an example of a looks like 100% of you put A. That's correct. This is probably the one example of the keystone species you learned about in class. So this question covered 8.6 and 8.7. So look at those if you had confusion about this. That brings us to the end of the content that I thought was most important that we covered tonight. For further review, because tonight's lecture was an attempt to pack the entire course down into a sub 2-hour session, check out AP Classroom, probably the most relevant one. It's specifically tailored for your AP exams. Definitely check that one out first. Also, check out the 5Hive website at 5hive.org. We've got a bunch of other content created by students who have taken the exams before and scored a 5 on it. We create these content for you, but it's also very much still a work in progress. So if you don't see what you need on there, we're probably working on it. Also, generative AI like ChatGPT is definitely advanced enough to give you some pretty good practice on AP exams, but also be aware that it's not going to be correct 100% of the time. So definitely check yourself on that. And then always, as I've said at the beginning of the lecture, I always recommend Khan Academy. It's free. It's very good. Some of the diagrams in this lecture I've taken from them, so why not check them out? Did I also mention it's free? And then again, a reminder about filling out the lecture feedback survey. It's going to help us grow as a project, and it's also going to help me as a lecturer. You can also tell me what worked for you, what didn't work for you, what you might like to see more of in the next lecture, so on. So at this point, I'm going to open it to any other questions you might have. If you don't have any more questions, you are free to leave. Thank you for coming tonight, and good luck on the exam.